15 crore to set up a complete lab, that you would have good instrumentation, all the kinds of protein cleanup facilities, standby liquid chromatograph, sometimes a standby mass spec itself. So things cost a lot of money. And then in any translational work, you would like to take your solution right from the lab to the clinic, from the lab to the agricultural land, right? That is what a good translational work would be. Like, it's a test that can be done by a layman, a test that can be as easy as like those lateral flow tests that you use, like for COVID we had used one, pregnancy testing kits. So something as easy that can be done at home or maybe done at a very rural dispensary. That's what we aim at as a scientist. So one way of doing it is by microarrays. Now microarrays, I think the first thing that reminds you about a microarray is that it is something related to DNA. And of course it did start off with DNAs, but now we have all kinds of protein microarrays, and that's what we will be discussing. And we will also telling you, using a case study from the work that we did at IIT Bombay while I was there, at how you can use microarray as a diagnostic and prognostic tool. So if I ask you what are microarrays, what would be your answer? Anyone wants to volunteer? What is a microarray? What is micro? Small, right? We have microscope. So all kinds of micros we have today. Micro USB. So micro means small. And what is an array? Collection of
instances
of what?
Specific object, or you can say protein, genes.
Right, anything that is systematically arranged in a repeated manner, right? That's an array. So if we go to the definition, so these are very miniaturized platform, and what protein microarrays can, or any microarray can do, it can allow for simultaneous detection of molecular interactions, right? Here the word is molecular interaction. It can be gene expression. I think Professor Graham started off his AI ML lecture with showing you a microarray grid, and he will be also going deeper into it sometime soon. And so it can check for gene expression. It can also check for protein binding. And what you can do it, you can do it on a very small, I think all of you have seen a microscope slide, right? And it's very small. It's very handy. You can carry it with a hand. That makes microarray something which is extremely portable. You can take it in your pocket, and the work that you need to do is, it can be done very fast. So what typically a microarray will consist with, will be an arrayed series of microscopic spots, and each spot will contain a specific molecule. It can be any molecule. It can be DNA, it can be RNA, it can be protein, it can be cell lysate. And what you do it is you immobilize it so that it does not move around on a surface. Usually it can be a glass chip, it can also be a silicone chip. And it all started with people started designing microarrays of DNAs, and what they did was they put small fragments of genes, DNA, and they put it on a glass chip, and when these spots were exposed to a sample containing DNA or RNA, they would hybridize, not bind. So DNA or RNA will always hybridize, and this hybridization would lead to a generation of a signal, and based on signal intensity and the presence or absence of signal, you get an expression data set, right? So what you do is you can compare different types of RNA from different samples. You can healthy versus diseased can be one way. Like you have the RNA, entire RNA you spot from a cancer tissue, from a non-cancerous tissue, and that will give you at the level of transcript, that is, it will tell you what are the genes overexpressed and not expressed or underexpressed. One thing you need to remember these are always targeted assays. These are not by far discovery. Why? Because in most cases you know what is what in the array. So this is one challenge of a microarray, that you cannot do much of discovery work, except for a few things that we will understand very soon. It can also, you can find single nucleotide polymorphisms if you're working with DNA. So these kind of stuff you can do. You can compare the genomic hybridization, you can compare about the number of genes in a cell, and you can visualize the DNA, the interactions, because most of these probes will carry some fluorescence dyes tagged to it, Psi3, Psi5, Alexa fluors, and when you scan it, you get a signal, right? Red, green, blue dots. So why do we use it? Because we can use it to understand diseases, drug responses, developmental stages. We can also study various mutations. We can also identify genes or proteins linked with a specific condition. So these are the things that you can do with the microarray. But when we talk of protein microarrays, so this was a later development, and what people did was similar to DNA microarrays. They started printing small proteins onto a surface or a glass chip, right? And these proteins can interact with other proteins. And how these proteins interact? A very common interaction is antigen-antibody, right? So if you have antigens presented on a glass chip, antibodies will come and bind. You can do the reverse. You can print antibodies on a glass chip, and then you would see what are the antigens that are binding to it, and that can give you a protein profile. It can also give a functional analysis, and you can do a kind of a drug discovery approach. You can do all these stuff. And this figure, I don't know how, is it visible well, or should we, can somebody turn off that light? This one? Right. So I think it is visible to you. Right. It's fine. So these are grids. These are small grids. And if you look at it, these are very small dots present on this microarrays. And each of these dots might represent a protein or a peptide, or sometimes a cell lysate. So depending on the kind of arrays. So we will now move on to what are the major types of protein arrays. So the major types are primarily these. So these are analytical protein arrays that are used for, so these are all analytical arrays. So what you have antibody arrays, you have peptide arrays, you have antigen arrays. We'll talk about peptide and antigen very soon. But what is an antigen array, antibody array? You have, imagine you want to check a cell signaling pathway, and you want to check that for a particular pathway in the developmental stage of my animal model or cancer, you want to check all inflammatory markers. So what you can do is you can print all the antibodies against inflammatory panels. Maybe you have a panel of around 50 or 60 markers. If you do, and you have to screen perhaps hundreds of samples. Now think that each grid contains 60 spots, and each of these spots in duplicate, that means a grid of 120 spots. Each of these duplicate spots are primarily an antibody against a specific protein in that cascade of signaling. Now you take the entire protein cell lysate, you incubate this glass chip. In one go, you can do a sort of an ELISA or Western blot of all these arrays, of all these proteins. If you have to do these individually, that you have to do for these 60 proteins, it will be extremely costly, and forget costly, it might be extremely laborious, and then you would have variations, because perhaps for these 60 blots, it would take at least 4 to 5 months. Temperature conditions might change, your mood might change, your buffers might change, and the lysate that you took, that after repeated freeze-thaw cycles, even that condition might change, right? So you can do it in one go, in one shot. Then you have peptide arrays, these peptide arrays. Now what are peptides? Short fragments of proteins. Now suppose you want to check that if your protein of interest is trying to interact with another protein, right? You have some proteins, you want to do a protein-protein interaction study, right? So you can either do a peptide array, or perhaps you want to check for antibodies present against a specific protein, whether you are generating autoantibodies. Now what you can do is you print these peptides, and almost similar is antigen, you can print it onto a glass slide, and then you can do something that we will discuss later, something known as an epitope mapping. You can do an epitope mapping. You can exactly know which part of the proteome is actually being antigenic. So if you go back to your immunology basics, you would understand that the entire protein is never antigenic, right? There are specific regions in the protein that are antigenic, and we call it the epitope. Now if you want to design a vaccine candidate, suppose you want to design a vaccine candidate. This vaccine candidate cannot be the entire protein. Maybe you want to design a peptide vaccine. Because proteins by itself can break down, it is mostly always thermolabile. You would need a cold chain system to maintain that entire protein sense. But suppose you want to make a peptide vaccine that you can carry in a pocket. You can use it as a nasal spray. So in the nasal spray you have this peptide mix. Because they are peptides, they are far more robust, and all you need to develop or to administer a vaccine is just do 2 sprays in your nostrils, and those peptides will be absorbed in your nasal mucosa and will start generating an antibody response. But to select for those epitopes, you can use these arrays. Now another kind of array is called the functional protein array. This is actually very important when you want to study protein-protein interaction. You can also do an enzyme array. Suppose you want to check for enzymatic activity. What all enzymes are present in your cell lysate? Then again, you cannot store your cell lysate for a longer duration. Why don't you go for an enzymatic array, where you can have substrates, and each of these substrates are unique to the enzyme, and if the substrate reacts, it will create a chromogenic spot. And based on the presence and absence of the enzyme, or maybe based on the presence or absence of the activity of the enzyme, your spots will change color, maybe something from faint yellow to brilliant yellow. And these arrays can again be scanned, it can be digitized, and once it is digitized, you have Professor Wall who will help you to apply his magic of AI and ML to understand your data better. But you can do this for a lot more. See, the arrays help you in increasing the throughput of your experiment. Now you are not looking at one single cell or one single protein, or you can look at the entire cascade. You want to study glycolysis, and you can have all the enzymes that are involved in glycolysis. In fact, you can go for some secondary enzymes that are part of the upstream and downstream cycles, and you can have a complete understanding of how these cycles are processing going ahead. Something that we do is reverse phase. These are all normal phase, and just as in HPLC or chromatography, reverse phase means here you put the cell lysate or the tissue lysate onto the glass plot. Right? Now suppose you have to check 500 samples for the presence of one antibody, and you want to do it very fast. You are in a rural clinic where there is a breakout of a pandemic, and you do not have the money or the time to give 500 results. You take a glass chip, you take a microarray printer with you, you take their blood serum samples, spot it on the glass slide, and then do one check, and based on the arrays, it can tell for those 500 patients at one given time if they're having a specific antibody, or if you are checking for a specific biomarker. So these are called reverse phase arrays. So now just going deeper how this analytical microarray works. So how they work is, these microarrays are designed to capture a specific protein and then detect their presence. I think many of you would have heard of would understood something known as ELISA, right? It's something of a micro ELISA, that we have miniaturized on a glass slide. So what they do is, if you have an analyte labeled, so this is the antibody, and this is the fluorophore. Now, and these are the reporter antibodies, and this hexagon is the target protein. Now, this target protein will bind to it, and the concept of sandwich ELISA is what? That if the target protein is binding, then it would not allow the reporter antibody to bind. So a binding event will be distinguished by the loss of signal. So if you have a bright spot, means there was absence of the target protein, right? If you have a really bright spot, because reporter antibody could bind with this antibody, and because target protein was missing. Now if you have the target protein, it will sterically hinder the binding of the reporter, and then you would have a very low faint signal. So faint signal means your target protein was present. A bright signal means the faint signal was if it is a bright spot, means your targeted protein was absent. So this is how analytical microarrays work, and these are specifically designed to capture specific proteins, detect their presence, and also sometimes, if you can standardize it well, it can give you a concentration in a given sample. So this is again something that is subject to specific conditions about how you have done the study. And they usually contain antibodies, aptamers, and other protein binding molecules. And why they are used? Disease diagnosis. We have just discussed that you go to a rural village and you want to diagnose 500 villages. You want to do a sero survey for all the 500 villages in one go. You can do it rapidly at a micro scale at very low cost, because since you have scaled down the experiment, your consumable cost will also be reduced. So in India specifically, because most of you, I think all of you are from India. So one thing, always remember, in India, when you develop a technology, make sure it's cheap, else you cannot translate it. We are not a first world country who can afford to have a 5,000 or 8,000 rupees test. So ideally you should aim something between 10 rupees and 100 rupees. Each test should not cost more than that if you really wanted to translate it on a big scale. Also, you can do protein profiling. If you're not working on diagnostics, you want to work on your own cell cycle systems or your disease models, then you can check for the relative abundance of various proteins in different samples. That also gives, without using a mass spec, a fairly good picture of the biological process. One thing I think all of you must have understood by now, nowhere did I mention that you have to deplete your sample, right? You don't have to deplete your sample. The sample processing step in these microarrays is minimal. You have to just take, if you're working with blood, and we have worked for COVID, all we took the plasma, or rather we took the serum, and we diluted it 100 times, 200 times. So even if you have to take blood, you don't need to draw 5 ml or 10 ml. Even a 1 ml draw would be sufficient for maybe 200 microarray experiments. So that way your procedure, or your test is cheap, is non-invasive or minimal invasive, as well as translatable at a very low cost. Then we have something known as functional microarrays, and as we had discussed, they contain full-length proteins or protein domains or peptides, right? So if you're wanting to study a protein-protein interaction, or suppose you want to study if a particular domain of a protein is interacting, then you can break down the protein, you can make those proteins, immobilize it on a chip, and then do your assay. And each different domains will give you different signals using the same sandwich principle, right? So this again gives you much more flexibility to work, and it can be used for protein-protein interaction study. You can also do protein-DNA RNA interaction study. Now suppose you are working on a transcription factor. right? And you want to check, or you're trying to do a promoter analysis, and you want to check whether my specific. So these kind of proteomics technologies gives you this is not an alternative technology. This is actually a technology in its own right, and it gives you much more advantage compared to a mass spec, because mass spec can never tell you if a protein or DNA is interacting with each other. It can predict, but it cannot give you hard evidence. You can also test potential drugs, whether a drug is binding to the protein of your interest, and it can also provide some actions, like it can give you some action points that how if the binding is being there or not, which domain is binding. All these kinds of studies you can do with a functional microarray. So just to tell you what kind of functional microarrays we have a protein-protein interaction microarray. We have a protein-lipid. P shows protein-lipid. C shows protein and cell lysate. D shows small molecule binding. E shows substrate identification of PTMs, substrate enzymes. Then you have protein DNA, protein RNA, antibody specificity or serum profiling. So you see just one technology, but with the variations it can provide. And if you see, none of these studies could be done on a mass spec. And they are usually very cheaper. So again, to get into details of reverse phase protein arrays, as we had discussed in the very introduction, that reverse phase is when you spot your cell lysate and then probe for a specific antibody to detect the presence of a target protein. Now you have 1,000 samples. You are working on a clinical, you are trying to establish a biomarker. Suppose you have established a biomarker using mass spec. Fine. You have established a biomarker against mass spec, and you are telling that this protein should ideally be a prognostic tool or diagnostic tool for disease X. Now, one good way of you don't have now you want to try to reach out to your collaborators across India, across the globe, and you want to validate it. Now you cannot ask everybody for a sample and they would ship it back to your lab in India, and you will run thousands of targeted proteomic assays. What you can do rather, you can do a reverse phase. You can tell them that, okay, you print it on your chip, and this is the protein antibody I'm providing. So if this antibody is binding to your serum, means this specific protein has gone up or is present in the serum or plasma of the patient. That can be you can validate your biomarker. So sometimes you can use these techniques to validate the findings that you get on a mass spec. So of course, disease progression is one of the ways you can do that. You can also study disease progression. Suppose there are stages of cancer, stage 1, stage 2, stage 3, stage 4. Then you have diseases like COVID that has a non-severe and severe stage. All these things you can do. You can also study the signaling pathways in cells. You can study post-translational modifications. So, and then how does it look like? So, all you do is you take samples and tissues, lysates or serum or body fluids, also subcellular fraction. You make a homogenate, and since these are proteins, it won't usually stick to a glass slide. You can take a nitrocellulose membrane, and on those nitrocellulose membrane, you can just allow these molecules to be absorbed in a particular array pattern. One thing to remember is, whenever you design an array, you have to remember which spot you are putting what. So on your computer, you should have designed the array, and just like you work in a 96 well format, you don't label each of the wells, do you? Right? You know that A1 is this, A2 is this, B3 is this. So similarly, think of it like a 96 well. It's a 96 spot array on a microscope slide. Then you can probe with fluorescence labeled antibodies against these biomarkers. And one more thing that you can do, you can actually do a simultaneous detection. You can use 2 antibodies. So 3 antibodies, depends on the amount of money you have. And also when I showed you the first grid, now this is here also you have a grid. So sometimes you can do specific incubations on specific grids. So the first grid, you can incubate with antibody 1. Second grid you can incubate, and there are 8 grids and 16 grids that can be possible on a specific microscopic slide. And based on the grids, if you have 16 grids, in one go you can assay for 16 different antibodies. If you have 2 types of fluorescence markers, you can go for till 32 proteins. So this is, since we are talking of big data, these are actually more of a high throughput approach that you need to think. So we don't talk in a high throughput approach, we don't talk about one data set or 2 data points. The data points should be large. So here the patient number will be large, the number of samples, assays you're doing should be greater. Now, one interesting thing is the peptide microarray. So here what you do is, instead of full proteins, you can print small sections of the protein and peptides, and since you're printing specific regions of the peptides, each region of the peptide will correspond to a specific motif or domain in the actual protein. And the number of applications that you can do is, one is epitope mapping, which we will discuss very soon, and which was much of my work that I did at IIT Bombay. So we'll discuss this in detail. And you can also study how specific modifications like phosphorylations on a peptide might affect its interaction. So suppose you have a protein that can get phosphorylated at 12 different positions. And yesterday I think we were discussing that different phosphorylations might lead to different kinds of activity. So you can have all the permutation combinations of those domains with different phosphorylation in each domain, and you can try to understand how each phosphorylation is affecting the biology of the protein. And how do you, what do you do? First of all, we can do biomarker detection. You can presence of biomarkers. You can have autoantibodies or autoimmune disorders. Very easily you can screen. Then you can do autoantibody profiling. Again, for autoantibodies, I'm indicating autoimmune disorders. You can check for those profiles that, okay, these particular person is having autoantibody against these proteins. We can do viral and bacterial detection. We will discuss how we can detect COVID, and you can do epitope mapping. Also, you can do drug target identification. You can find, okay, this is the drug that interacts with this domain or motif of the protein. You can personalize treatments based on these profiles. And also you can screen a lot number of drugs using this, because you are using a high throughput approach. You can screen multiple drugs, how they're interacting. So multiple studies, you can do interaction studies, you can do post-translational modification studies. You can also, as we discussed earlier, you can also try to find what are the probable or the good vaccine candidates, right? So now we will see how we used this microarray while we were doing our studies on COVID-19 in India. So this is the general trajectory of what happened during COVID. So what happened was viral particles entered into our airways, and the presence of SARS-CoV-2 receptors, the ACE2 receptors was actually the ACE2 protein that was present on the cell cycle, on the surface of the lungs. They allowed the virus to enter so they allowed the protein to interact, this virus to interact, and they got internalized. Then the mumbo jumbo of immunology, we had the antigen presenting cells and the helper T cells. It helped and activate, and the macrophages and the dendritic cells, all of them together led to the activation of the helper T cell, which activated the B cell. This B cell started producing the plasma cell. This plasma cell again produced antibodies, and of course there is a class switching event. So initially they produced IgM or IgA, and then it produced IgG. Now what we could do is, using these epitopes printed, or these peptides printed onto the glass slide, we can see which peptides were actually antigenic. The entire virus or just like any other protein, the entire protein can never be antigenic. So what did we do? So this was something that we purchased from a company called Pepperprint in Germany. So what they did was they printed all the peptides present based on the Wuhan original sequence that was sequenced at Wuhan in China. They translated into the viral proteome, and the translated proteins were printed in an overlapping manner. You see, the first amino acid was 1 to 15. The second was from 3 to 17. So there was a 13 amino acid overlap. So now you're creating a grid where each of the peptide has a 13 amino acid overlap. So I want you to visualize, if you do this overlap, you have a very high resolution of it around 2 amino acids. The resolution that you're achieving is of 2 amino acids. 15 amino acid peptide, and you have a 13 amino acid overlap, which means this grid is actually moving 2 places at a time. And all the peptides right from nucleoprotein, spike protein, ORF3a, envelope, membrane, ORF1ab, ORF6, 7a, 8, 10, all these were printed onto a glass slide, and that led to 4,883 spots multiplied by 2, because all these spots were in duplicate. So these number of peptides were printed onto one glass slide. Of course, it came at a cost. It costs us around 65,000 rupees for one slide. But what we did was, we wanted to ask a biological question, that does severe and non-severe patient have a differential expression? Does their immune response vary? Right? So what is it that few patients getting infected by the same coronavirus is having a very extreme severe response? Somebody is having just a normal response, usual response. So is it their immune response in the first place? Is it how their immune system was latching onto the peptides due to neutralizing antibody response or binding antibody response? So all this we wanted to study, and for that we'll go to the linear. So what we did was a linear epitope mapping. So this is a linear epitope mapping, and if you clearly see here, so these are overlapping peptides, right? So this was the entire SARS-CoV-2 protein, and you see there is a fair amount of overlap by the way the grids are kept. These overlapping peptides were printed onto a glass chip, and the IgG or IgM as possible were started to bind. Now imagine a specific antibody that is binding from, suppose antibody, its binding site is between amino acid 4 to amino acid 18. That is the motif. So it will very weakly, but surely it will bind with the first, because there is an overlap. It will also bind with the second, because there is an overlap. Now this binding will create an array of spots, right? And since we have printed in duplicate, it will be a double spot. When there is a double spot, you would, via data analysis, we could actually look into the specific consensus motif. The specific consensus motif. So this is the binding pattern, and this binding pattern will help me identify with a high resolution of only 2 amino acids. Only 2 amino acids. This will give me a very high resolution about, okay, this is where the protein, the antibody is binding. It can be IgG, it can be IgM, it can be IgA, depending at what you want to study, right? So this is what the grid of IgA used to look like. Now you see, this paper is already published, so in 2023. So this was what the scan looked like. Now if you look very carefully, you will see the entire slide is not stained red. You can see small dots, right? So these are all double spots, and when you zoom in further, so this is the overlay of the grid. So the computer, so the guys who have printed this, they already know which peptide is present at which spot, right? When we overlay the scan with this grid, we know the signal is coming from which peptide. And based on that, what we found, if you look at it, I don't know if it is visible, so this is the severe group, the severe patient. This is the non-severe patient. Now in severe, you find a very strong expression, whereas in the non-severe group, you find a very weak expression pattern. This is just a representative, and this is how we plotted it on a slide. Similarly, again so you find the severe group, at least in this IgA channel, initially mounts a very strong antibody response against ORF1ab and these specific regions of the protein, right? So these are ORF1ab protein, this is also ORF1ab, and I think this is the glycoprotein, membrane glycoprotein, this is also ORF1ab protein. You can go to this paper and you have all the data sets available. So this is how we wanted to understand if there is a we could distinguish the response given by severe and non-severe patient. Thereafter, we moved on to the IgG response. And here also, we can see that in case of severe, and this is this classical double spot. You see, I think this is very clear. So this is a very strong binding. This is for nucleocapsid, this is ORF1ab polyprotein, this is for nucleocapsid, this is for ORF8 protein. So again, you can see this grid. So this entire slide did not have this green background. So these are specific spots, and all of these spots, if you go back to the literature of the manufacturer, we can exactly tell what these spots are. And what you see in these boundaries is the control, and what could possibly be the control. So this checks for polio. It's against polio virus, and all of us are vaccinated against polio. So we would have a strong response for polio for all the patients, but this is only for SARS-CoV-2 proteome. So this gives you an idea about how you can use microarray. So I told you about different forms of a microarray, and I also showed you how you can. So these targets, these are these protein targets. We can easily say, if we suppose take this protein target and we tell that if there is antibody response against this specific peptide, a clinic can usually do an ELISA, right? We can convert it into an ELISA target. A clinic can also run a microarray, but perhaps they don't have a scanner. Microarray is also costly. Slides cost 50,000, and the scanner that we used was around 2.5 crores. So yeah, omics is actually costly, because that's mostly because there is very less adoption of the technology. More and more, you go back to your labs, you convince your supervisor, and you have more sales, the prices come down. So that brings us to the completion. So you can just go through this paper. I will share this paper at the end of, as I said, few of the papers that we have been referring to. I will also ask for some papers from Professor Gramball, some reading material that you can read through. Right now, if I even share the reading material, you hardly have the time, because we are moving from one topic to the other. So once the paper is done, I mean your entire course is done, you can go through these papers, these folders, study material on microarrays, study material on targeted proteomics, study material on MetaboAnalyst. So all these tools that we are discussing with you, Reactome, MetaboAnalyst, they have their own individual research papers. These are developed by research groups. You can go back to those papers, read them. Most of these tools would also have their own tutorials that you can actually spend 4 to 5 days. So there are Max Quant summer schools. I think that runs for almost 5 days. So of course, whatever they will teach in 5 days, we could hardly cover in 1 hour. So the idea is to give you a flavor. Things exist. You know Max Quant exists. It's free. You can use it in your laptop. perhaps if it is having a 16 GB RAM, but mostly you don't have to pay money for it. So that brings us to the end of this session. If you have any questions, and this is why you should attend this GAM program. So the fire needs to be kindled, right? We are not aiming to fill you with information. We'll give you more and more things to choose from, and then whatever you feel like you want to carry on forward in your own research, in your area or domain of expertise, pick and choose. So anybody has any questions? I think we can take a couple of questions at most, and then I will hand over the stage to Professor Graham so that he can continue the proceedings for the day. If you have no questions, I feel you have not understood anything.
Since most of the applications that you told us about the disease identification or drug delivery,
Right.
so don't they have any application in plant and animal studies? And
Plant and?
animal studies? Different from diseases and drugs, I would say.
As I said, when you study cell cycle, right, your plant would be having lots of, so those are most of these studies that since I have been working on animal models and diseases, so I picked up examples. Of course they would have it for plants, so plants would produce proteins, and proteins would obviously react with antibodies, but using those you can make your own arrays. I mean, you can, there are companies like Pepperprint and Ray Biotech, I think. In India you don't have it, you have to go abroad. You have to talk with them. They can make you custom chips, but you can do it. It's not impossible. If it is a protein or a peptide, it will definitely bind with someone or the other. You can do DNA protein interaction studies. So if I go back to this slide, so you see there are so many interactions, and this interaction can be translated to algae or plant or animal or to a disease. So it's up to you. So that's how you, as a scientist, you have to wonder what possible thing you can use. If you want to use as it a protein lipid interaction, you can go for customized microarray printing. If you do it in bulk, of course it will be cheap. If you ask for one chip, of course it will be extremely costly. But things can be done. Anyone else?
So what scanner exactly is used?
So there are arrays, scanners like GenePix is one of the scanners. You also have this scanner. This is from Inopsys. So the photograph is from Inopsys, their website. So Inopsys is one scanner. GenePix is one scanner. If I'm not wrong, Agilent also makes a scanner. So the reason they are very costly is because they use lasers that are these are monochromatic lasers. This is the laser I'm using is very cheap, because if you look at the wavelength, it might look green to you, but these are not perfectly monochromatic. So monochromatic lasers, one reason why they are costly. The camera that they use, you get extremely high resolution. You see, these are microscopic slides, but when you get the actual image, it's almost like 45 MB file, and you can zoom into 2-micron dot, and it appears like a circle or a bar. So that drives up the cost. But again, if you do the work in collaboration with a lab that already has a scanner, it brings the cost down, right? And in India, I think there are almost 5 or 6 scanners that I personally know. One is with Professor Sanjeeva, other is I think at NII Delhi. One is at Thisti, Faridabad. So I'm not very sure about these things if they're still working. But when I was wondering, so you have one Agilent instrument in BHU itself. So you can do that. I mean, you have to plan your work. You can do your incubations, and you can just scan them. And these slides are very robust. Since it's a glass slide, mostly you can carry it in your pocket. It does not need a dry ice shipment. And most importantly, for human studies, you don't need to deplete your plasma sample. That's one of the big advantages, that you can save a lot of money on sample processing costs. No buffers, nothing. All you need is physiological saline and saline and few of these buffers that you usually use for Western blotting. Our blocking agent, I did not go into the protocols, but we can share with you the protocol as well. This paper, though, I was mentioning, it has a protocol. Okay, anyone else? Or I can wind up and I can hand over the stage to Professor Graham. So thank you for your patient listening. Professor Graham, welcome back. The stage is yours.
Thanks.